Hello everyone, I'm Ayako Kamei, Senior Tech Support at OriCiro Genomics. Thank you for joining today's webinar. I'm happy to have a talk today. Today I'm going to talk about our cell-free cloning system and our products too. This slide is a typical way to amplify the plasmid DNA using E. coli system. So let's say you have a plasmid DNA, only plasmid DNA, like maybe you received from your colleagues or you made it by yourself. So when you have your plasmid DNA, so what you do first is at day one, you will do like E. coli transformation. And day two, you will see maybe at the morning, you'll see a lot of colonies on the plate. And when you see the colonies, you will pick them, maybe few colonies from the plate, and then pick the colony and inoculate it into the liquid medium, like LB medium with some antibiotics. And once you prepare that liquid medium, you will incubate it in with shaker and put into 37° and culture for like overnight. So before you go back home, you like inoculate the colony and then leave it into the incubator. So day three, next day, this day will be a little bit busy day. You might extract the plasmid DNA from the culture medium. And when I was graduate students, I learned from my boss that to make the glycerol stock too, not only extract the DNA, but make the glycerol stock just in case. So usually I prepare like two DNA, plasmid DNA and the glycerol stock. Also like two tubes. One is for -20 refrigeration, and then the other is for -80 refrigerator. So this is a really typical way to amplify your plasmid DNA. And E. coli is very powerful tool and everybody uses it. So very nice tool, but it takes a little bit time and you have to work a little bit more. And in my case, like I have to prepare not only DNA, but like both like glycerol stock for backup. So you need a little bit more time to prepare both. And especially like in like company industry, like there are very strict and they have to make a lot of documents or like a biosafety issues. You have to concern about these issues. And also, like when you're working with plasmid DNA, if your plasmid DNA is smaller size, then that's okay. But like if you have to work with like a larger size, like 50 kilo base pair, then the mini prep is not working well. So you have to do a little bit more work to get your plasmid DNA. So I know it's like E. coli is very nice strategy. Using E. coli is very nice strategy, but it takes a little bit time, and you have to think about like biosafety things too. So our technology is cell-free, so you don't have to work with E. coli. So no transformation, no liquid culture, and no glycerol stock. So our system is simple. So when you have your plasmid DNA, you need some magic powder to your plasmid DNA at day one. But once you have that magic powder and mix with our OriCiro's amplification technology, incubate it for 33° for 6 hours and just wait for it. And then next day you will have you'll get your amplified plasmid DNA. So no glycerol stuff, but you already have a plasmid DNA. So what's happening on this day one? The key point is E. coli's DNA replication system. So this slide shows the system of DNA replication in E. coli. The left figure is diagram illustrating initiation of replication of the circular chromosome at oriC and termination at TER. So oriC is the origin of replication of E. coli. The chromosome replication of E. coli initiates when the DNA protein DNA binds and open the duplex DNA at oriC. So the ribosome complex showing here, this complex replicates the chromosome in opposite direction from oriC, with each synthesizing new strands. So the duplication of the entire chromosome is completed when these two forks meet it in the terminus region, about like approximately halfway around the circular chromosome. Well, under the conditions allowing rapid growth, each of the duplicate origins can fire again before the previous replication forks have reached TR. So this system is very important for our OriCiro technology. So we have two key points of amplification technology. Number one is oriC sequence. This is the identity of the magic powder. So this magic powder is a double-strand DNA which contains oriC sequence. And the second is proteins. So in our amplification system mixture, it has like 26 proteins are in one tube. So these 26 proteins are involved in E. coli's chromosome replication system. So these two is necessary to use our OriCiro's amplification. So what is the 26 proteins? So these are the list of 26 proteins. And here, there are 3 proteins for initiation part and 15 proteins for fork progression, 3 proteins for Okazaki fragment maturation, 1 protein for termination, and 4 protein for decantination. These all 26 proteins are in one tube, and this mixture allows you to amplify your plasmid DNA by cell-free. These 26 proteins are expressed in E. coli cells, and each proteins are purified from the cells. And this 26 proteins is already mixed in one tube with like really nice cocktail to allow the DNA amplification. So this mixture allows you to incubate into the 33-degree for a few hours for the amplification. The amplification, this rapid amplification and exponential manner, like here you can see here. And then these amplified plasmid DNA will be the next template DNA for the amplification. The only thing you have to remember, remember is like only oriC sequence. You need the oriC sequence to amplify and to use our cell-free amplification system. So the magic powder, you need the magic powder. So what you can do for our system amplification system is when you run our system, you will get up to like 100 nanogram of your circular DNA per microliter reaction. It's from a single DNA molecule, and in 3 hours for like 33 degree, and you will get this amount of DNA. But it depends on the yield. It depends on the DNA size. If your DNA is smaller, then you may get like almost close to this number. But like if you're working with like longer DNA, then the yield will be a little bit low. And also up to 50 kilobase pair size of circular DNA can amplify using our OriCiro system. So our system is so cell-free, so we're not using E. coli. So if your DNA is something cell toxic, then that's not good for E. coli, but our system is cell-free. So it's okay for using your cell toxic DNA for our system. Same as like ZC-rich, a little bit difficult DNA like ZC-rich, AT-rich, or repeat sequences. These sequences you can use in our amplification system. Maybe I told you before that like the amplified products can use as the template DNA to amplify it again. So what you need a fresh reaction mixture, but you can use your amplified DNA to produce more plasmid DNA. So that's why you don't have to prepare the glycerol stock. Again, I know I'm telling so many times about this, but you have to prepare oriC sequence to amplify your plasmid DNA using our system. This is my data, like in-house data for using our OriCiro system. We amplified actually assembled and amplified, but amplified 8 kilobase pair and 50 kilobase pair of circular DNA using our system. So we prepared about 8 kilobase pair or 50 kilobase pair of linear double-strand DNA, and then prepare each oriC sequence which fit to 8 kilobase pair or 50 kilobase. Like assemble with 8 kilobase pair or 50 kilobase pair. So prepare like each oriC DNA sequence for it. And then when I use our system, assembled oriC sequence and then 8 kilobase pair or 50 kilobase pair DNA fragment and see how it works. Here, for 8 kilobase pair, you could see the nice, beautiful band at almost expected size of 8 kilobase pair. It's supercoiled DNA. So this ladder is supercoiled DNA marker from the second band with 8 kilobase pair size, and it's almost same position. You see this band. And here you can see two bands here. One band here is open circular, and the upper one is a concatemer. Same as 50 kilobase pair, I've also done the same thing as 8 kilobase pair, and I got the band of 50 kilobase pair of supercoiled DNA. So our system can amplify like up to 50 kilobase pair size of circular DNA. These DNA, of course, have oriC sequence. So like, how should we make the circular DNA with oriC? So I guess some people has only the DNA fragment, or maybe some people has like already have a plasmid DNA to amplify in E. coli. The first thing is you need the oriC. So you need a magic powder to your plasmid DNA. To prepare your plasmid DNA as a linear DNA, there are several ways to do. So one way is using restriction enzyme. So find the nice site that not cut your DNA, but cut your plasmid DNA backbone and with no effect to other genes. So find the restriction and good restriction enzyme site, and then just digest it with that enzyme. It will be the linear. And better to purify that DNA to remove the enzymes. If you don't have the if you couldn't find the good restriction enzyme site, then CRISPR-Cas9 will be the second way to make a linear DNA. Also, the third way is the PCR, design the primers and use your plasmid DNA as a template DNA and then just do the PCR. So you need the linear double-strand DNA to assemble with the oriC DNA sequence. So once you prepare your long DNA, then you also have to prepare oriC DNA fragments. This oriC DNA fragment should include like 40 to 60 base pair overlap sequence with your DNA. Once you prepare like these two DNA and then just use OriCiro assembly system and make your plasmid DNA, your DNA with oriC sequence, and you will get the circular DNA. Sorry. And use this, once you got your plasmid DNA with oriC, then use amplification system to make more of your DNA. So the OriCiro assembly system is not super specific to technology to us. There are so many companies that like have these kind of technology like Gibson assembly or infusion. So it's not so unique technology for us. Now, so when you use our assembly system, it's like similar as other technique. Like you need the overlapping. You have to design the overlapping ends to each other, the end of your DNA fragments. So when you mix with our assembly mixture, the one side of the double-strand DNA is digested with nuclease and exposing the single strand containing the overlapping sequence. After that, another enzyme comes to your DNA, like single DNA part, and then anneal each other. And here you can see some gap or maybe a nick after the annealing, but it's okay, just you can leave it here because in our amplification system, there is a ligase in that mixture. So that ligase will fix your DNA during the amplification reaction. So for assembly, our technology assembly, it's like you need like 40 to 60 base pair, or maybe sometimes it works with 25 base pair overlap, but just one step assembly if you have like 40 or 60 base pair overlap fragments for each DNA fragments. And you can assemble up to 50 DNA fragments into a large circular DNA with a single step enzymatic reaction. So our assembly is not heat reaction, it's enzymatic reaction. So it's more specific than heat reaction, we think. It's same as amplification step. Like it's on assembly is also it's isothermal process. So to summarize the advantages of our technologies compared with PCR, E. coli cloning system, and in our technology. So DNA size, like E. coli cloning system, our size is up to like 50 kilobase pair. The PCR, 10 kilobase pair, I guess more, you can do more than 10 kilobase recently. But like if you work on longer DNA, it will be a little bit difficult to work with PCR. Duration is, our technology is very simple, like I showed, and it's compared with E. coli system, you can save your time clearly. For PCR, I think yes, it's also simple, but when you think about the primer design, and you have to think it carefully. For biosafety part, PCR is cell-free and our also technology is cell-free. We don't use the E. coli system. That's a really nice point. And maybe the fidelity is very important thing to compare with these technologies. So compared with PCR, our system's error rate is lower than PCR. However, like compared with E. coli cloning system, it's a little bit behind than E. coli. So the one reason is in our amplification system or an assembly system, we don't have a mismatch repair protein in assembly or amplification. We think that's why the error rate is a little bit behind than E. coli. So we're now trying to catch up this part to find the good protein and then mix with to in our system. Sequence about the sequence. Our sequence is so our system is okay with cell toxic sequence or GC80 or a repeat sequence. PCR sometimes it might be difficult to amplify the GC reach or repeat sequence. So compare with that, like our technology can amplify any sequence. And the products are PCR linear DNA, but the R system is circular DNA. So there are technologies that amplify the DNA with cell-free, but it's as far as I know, it's most of the technology is amplifying the linear DNA, but our technology is amplifying only the circular DNA, which has oriC sequence because we're using the equalized chromosome replication system. Oops, sorry. So I will show you a little bit about data, in-house data about our system. So this is assemble and amplification data with GC-rich DNA fragments. The Sermis thermophilus is a bacterium isolated from a hot spring. And so this bacterium has a plasmid DNA, which size is 9.3 kilobase pair, and the GC content is 70%, pretty high. When we put the oriC sequence into this plasmid DNA using our system, and then we've got like a very nice plasmid DNA here using our system. This is PTTH plasmid marker, and then almost same size, because like oriC sequence is about 0.4 kilobase pair. So almost same size, we got the plasmid DNA was amplified. So even if GC rich sequence, our system can amplify the plasmid DNA. Also same thing as for AT-rich sequence. So we use mycoplasmas, mycoides, then genome DNA to part of genome DNA, and then try to amplify the DNA fragment, AT-rich DNA fragment with our system. Well, I prepare like 3 kilobase pair size of AT-rich 75% DNA fragment and 7 kilobase pair of the AT-rich is 79% of DNA fragment, and prepare the oriC sequence which fits to 3 kilobase pair or like for 7 kilobase pair. And after like assemble and amplification using our system, I got like 3 kilobase pair size, DNA size band, clear band here, and also concatemer here. You can see here the band here, but once you treat it with exonuclease, it will gone. So you can get very clear 3 kilobase pair of supercoiled DNA. Same thing for the 7 kilobase pair. Here you can see some open circular and concatemer here, but the most main band was the almost 7 kilobase pair size of supercoiled DNA. And after, same as 3 kilobase pair, after exonuclease treatment, the concatemer were gone, and then the main band went to 7 kilobase pair supercoiled DNA. So GC rich sequence, AT rich sequence, when you use our system, you can amplify DNA. Well, to tell the truth, 7 kilobase pair supercoiled DNAs is a little bit unstable. We have several times that we definitely we successful have a successful this 7 kilobase supercoiled DNA amplification, but it's a little bit hard to find the really nice amplification condition. We're now trying to figure out um, the the way to get more stable amplification for 7 kilobase pair size of AT-rich sequence. We want to make it more longer amplification for AT-rich sequence. Also, this is for like multiple fragments DNA assemble and amplification. We prepared 51 fragments, DNA fragments about the size is about 600 base pair, and each fragments have 60 base pair overlaps. To prepare these DNA fragments and put into one tube, mix with our assemble system, and then directly go to the amplification system, and we've got like 27 kilobase pair supercoiled DNA here. So even in 51 fragments, we can get the plasmid DNA with using our system. So this is kind of summary that like using our technology, you can save your time and you don't have to work with E. coli. And using our system, well, maybe you can make like viral or fetch using with these kind of genomes, and you can modify or construct the new organism maybe. And or the good point is like our system can work with large long DNA, which is maybe might be difficult to amplify the PCR, but if you have some fragments like a very long one, you can try our system to amplify or amplify it. Cloning for DNA, which is difficult to amplify in E. coli, our system is cell-free. So even if your gene is cell toxic, then that's okay. You can try with our system because of cell-free. Like I show you the data that GC-rich 70% GC-rich plasmid DNA, or like about more than 17% AT-rich DNA fragments. Circular DNA also that kind of DNA is also available to, is okay to using our system to amplify your plasmid DNA. Okay, so thank you for your attention. I'm happy to take your question.